Elizabeth Ryan, and I'm going to tell you a little bit about a condition that everybody's experienced, okay? Diarrhea. Everybody's had it, right? I mean, it's horrible. It's horrible. And you don't want to have it very long, and you know it's going to end, and you're going to be okay, right? I mean, but in the developing countries, this is not the case, and we've known this for decades, okay? And we've come up with solutions, water sanitation, we've tried oral rehydration salts, we've done all kinds of environmental cleanup. I mean, we're still having this massive problem, particularly in children under the age of 5, and particularly when you have these conditions of malnutrition. And so, we need to come up with some new solutions. And so one of the ideas came back from something I'd heard about, which was in a small part of India where there was, uses of rice bran. And so, as Jan mentioned earlier, this is a byproduct of rice processing. 70 million metric tons of rice bran are produced every year. Most of it is largely wasted and used for animal feed, though. I mean, now more and more, I'd say, as we go around the country, we know it's being used for animal feed. We just started in the laboratory, though, about 4 or 5 years ago with money from the Bill and Melinda Gates Foundation to do some preclinical work in animal models and just see how effective is it for Salmonella, E. coli, rotavirus, the leading causes of gastroenteritis in children. And we've had some great results. And so I'm proud to say now that we've been developing rice bran as a food ingredient, and we're feeding infants and young children in places like Nicaragua and Mali and West Africa to start. We're hoping to expand these studies and find out if there's some opportunity to really prevent and address this diarrheal disease condition, and also simultaneously address some of the malnutrition that's making it really a major cause of morbidity and mortality today.
So thanks. My name's Greg Ebel. I'm the director of the CSU Arthropod-Borne and Infectious Diseases Lab. And I want to start by directing your attention to the figure that I stole from Bill Gates' blog on the left side of the screen that lists the impact of the most deadly animals in the world. What should be immediately obvious to you when you look at that figure is that the most deadly animal on the planet is not a shark or a rattlesnake or a lion or a bear. It's a mosquito. They're even more deadly than human beings. So the reason that they're so deadly is that when they take a blood meal from you, which they need to do in order to complete their life cycle, they have the opportunity to inoculate viruses, parasites, and other pathogens. And there's a lot of great examples of these. We heard about West Nile virus already. Chikungunya virus and dengue viruses are persistent problems in the tropics. And this is to say nothing of malaria, which impacts millions of people each year. So the Arthropod-Borne and Infectious Disease Laboratory at CSU, as Sue mentioned, is 9 faculty. We're incredibly collaborative and integrated, and our goal is essentially to make that red box smaller. That's what we're trying to do. And so in order to do that, the faculty that are part of our group have both basic science and applied projects. Some examples of the basic science that we're doing include studies of RNA virus evolution, mechanisms of insecticide resistance, all kinds of different projects. We also have a lot of applied projects that tend to be towards vaccine development, finding new uses for old drugs. There's a picture of one of our faculty members at the bottom center who's using ivermectin to help control malaria in Central Africa. We also are very excited because we're thinking about new ways of using mosquitoes. And so for the last few seconds, I only have 2 minutes and 30 of them. I want to just tell you about a project that we're really excited about because we're thinking about using mosquitoes in a completely different way. So the idea here is that mosquitoes are biting people in resource-poor parts of the world all the time, and they're doing it anyway. And the idea is that in those mosquitoes, there's blood, and that the blood has information about the health of the people. And so what we're doing is we have developed collaborators at the Department of Defense and in Liberia and throughout the world. And we've been very fortunate to be supported by the Defense Department and by our CSU VPR. We take the mosquitoes that have fed on people living in huts, we bring them back to the lab, squeeze out the contents of their blood, of their midguts onto filter paper, which preserves the contents, and then we can bring that back to the lab and analyze it. And why we're so excited about this is we just recently have been getting some new data, and it seems like it actually works. So not only is it a new way of using and looking at mosquitoes, but we think we're actually going to be able to use this for biosurveillance.
My name's Dick Bowen. Virtually everything I do is collaborative. The thing that gets myself and my students, most of my students, up in the morning is the study of the so-called emerging infectious diseases. Now, that term's used a lot, and really what it kind of means is pathogens that are newly recognized or are showing up in new spots around the world. So the Ebola outbreak that we witnessed recently is still considered an emerging infectious disease. So my primary interest is in the animal reservoirs for these emerging infectious diseases. And as Sue kind of alluded to, that pie chart on the first slide indicates an important point, and that is that it's crystal clear that roughly 2/3 of these deadly, if you will, emerging infectious diseases of humans originate in some kind of an animal reservoir, and half of them actually in wildlife. So as people encroach on wildlife territories more and more, we're coming in contact with their pathogens. Next slide. So the virus I thought I would tell you about is a truly emerging infection, and that's MERS coronavirus. So MERS stands for the Middle East Respiratory Syndrome virus. This was first recognized in 2012 in a patient that died in Saudi Arabia, and it has since been recognized in a number of those Middle Eastern countries. Of course, with global transport systems as they are today, it was inevitable, and it has occurred, that people travel to the Middle East, pick up that virus, go back to their own countries, and spread this virus across the world. Sue also just mentioned the recent or the ongoing outbreak in South Korea. As of this morning, there have been 122 confirmed cases of MERS in South Korea and 9 fatalities. So the big question when MERS was first discovered is, where is this coming from? And most people, myself included, assume that there's got to be some kind of an animal reservoir. The two species high on the suspect list initially were bats, insectivorous bats, and camels. And there's part of our capability at CSU, we have the facilities and the expertise that allow us to safely manipulate these viruses and look at their effects on natural hosts, a variety of natural hosts. And so we started looking at bats and at camels. The story in bats is an anticlimax, really quite boring. If we inoculated 2 species of insectivorous bats, granted they were North American bats, with this virus, and saw no shedding, no disease, nothing. There hasn't really been any field data to support the role of bats in the epidemiology of MERS. Camels were also highly suspect, and we, as you see in that one picture with one of my graduate students, she's not really that small. It's just kind of the scale. We don't use children in these studies, but we inoculated dromedary camels with this virus. They developed an extremely mild respiratory disease that might even go unnoticed, but what they did was shed huge amounts of virus and nasal secretions. The amounts of virus that I think it's just obvious that would infect humans in contact with those camels. So what we're doing now to proceed from that particular study is we're collaborating with National Institutes of Health and some other groups to test vaccines in camels. The idea being that if we could develop a vaccine that would protect camels and decrease or eliminate shedding, then that would be useful in protecting human populations.
So my name is Diane Ordway, and I'm an infectious disease immunologist. And so it's one shocking fact is actually there's an extra 10 million people this year that will lose their lives to drug-resistant infections. And so it's one thing to read this in a report, but it's quite a different thing to actually work with patients that have drug-resistant infections. And so as you see on the slide, this is Miguel. He came into the hospital Agues Muniz in Lisbon, Portugal, where I was working. And he was diagnosed with Mycobacterium tuberculosis. You can call it TB or tuberculosis, which Miguel got this infection from somebody else that was coughing it up, and he breathed it in. And so he had a drug-sensitive infection, and what this bacteria does when it gets into your lungs is it basically destroys your lungs. Obviously, you need your lungs to live. So it's very lethal. And so he had a drug-sensitive strain of Mycobacterium tuberculosis, and so basically the clinicians gave him a standard drug treatment regime of 9 months and sent him on his way. Unfortunately, what happened is Miguel, about 6 months later, showed up in the clinic coughing up blood. So what Miguel did is he started to feel better on the drugs. He stopped taking the drugs. And then the infection came back, the bacteria changed. And so in the clinic, he had acquired a multi-drug resistant infection, which is very complicated because he ended up having to take more toxic drugs that cost a lot more money and for a lot longer time. So he inevitably developed various complications to these more toxic drugs. And although he fought a really good battle for 3 years, he lost his life. And so really, it's quite different, as I said, reading a report and actually working with patients with drug resistance. My research is really focused on getting new, non-invasive, point-of-care diagnostics for patients like Miguel and patients all around the world. And it's based on breath, exhaled breath. So using breath as a rapid diagnostic parameter. And so what we're doing at CSU is actually using exhaled breath. We're breaking down the components of that exhaled breath into molecules. And so we can detect from breath, because there's a molecular shedding of your blood through your lung cavity, of any type of infections you have, so we can identify that infection, the specific infection, and then also identify if that infection is drug resistant or drug susceptible. So we're using discoveries with our preclinical modeling, and we're also trying to increase efficacy, obviously, in the public human health sector. If you see the slide where the individual is taking a breath sample, it's much like asthmatics do routinely in this country to monitor their asthma, and also much like the alcohol breath test. So we're really wanting to shove this technology so we can use handheld breath analyzers, not just these large current equipment that we use to analyze breath, but also using a lot of Chuck Henry's sensor arrays so that we can actually have positive and negative readouts of whether an individual is infected with a certain pathogen and if it's drug resistant or not.
I'm Rod Page. I'm the director of the Flint Animal Cancer Center. The Flint Animal Cancer Center is the largest center devoted to the understanding and treatment of companion animal cancer in the world. We have about 25 faculty and about 100 people altogether across campus that are involved with basic and translational research, both for the benefit of animals and humans. We've been in existence for probably about 35 years and have been funded through that time period by federal and corporate and philanthropic sources to conduct research that looks at some of the fundamental opportunities to integrate cancer research and treatment across species, in addition to managing the patients that walk through our door. These are animals that develop cancers very spontaneously over the course of their lifetime. They're owned by individuals that come from around the world to see us because of the technology and the expertise that has developed at CSU. There's about 1 million dogs and about maybe 500,000 cats a year that get cancer. In comparison, there's about 500,000 people a year that get cancer. So our premise is that cancer is cancer. The more we look, depending on what sort of platform you want to use, we can't distinguish a cancer in a dog from a cancer in a human. We can certainly distinguish the dog and the human, but under the microscope and from an expression analysis platform, we can't tell which is which. They randomly assort. That gives us opportunities to investigate new types of treatments and targets that might be available that will come along. A couple of examples of some of the things that have happened here over the last couple of decades. On the left of your screen is Emily Brown, who was diagnosed with osteosarcoma of the spine when she was 10. Through research that we conducted and through the cooperation with her pediatric oncologist at Children's Hospital in Denver, we identified a particular compound that resulted in her being able to overcome some of the resistance mechanisms that allowed her to continue, and she just a year ago graduated from college. On the right is Dr. Steve Withrow, who's a surgeon, an oncologic surgeon, who developed a procedure to save the limbs of dogs that get bone cancer. That is currently the standard of care for kids with bone cancer. And Angela is on the left, and Angela has just turned about 17. So these are the opportunities that we have. Next slide, please. What we think we can do is to create an integrated mechanism where we can utilize the patients that we see with the human patients that walk into an oncologist's office to better accelerate the pace of cancer drug development. It's true that over 90% of cancer drugs that are initiated in the clinical trial pipeline never make it. That's much higher than in other classes of compounds. One of the reasons is that the traditional rodent species that are used and the other normal types of evaluations are just not predictive of the cancer situation in humans. The patients we see are similar. They're usually elderly dogs and cats that have concurrent issues that are on medications similar to human patients might be, and we found that we can help to predict whether a particular drug is going to be particularly tolerable in a human, much better than a rodent or a dog, normal dog's treatment process. But we can insert the system throughout the process of drug development evaluation. So we believe that the answer to cancer is usually walking right beside you.